The word cancer evokes fear in our mind, and why not? It is a disease that claims more than millions of lives annually in India alone, and has been the subject of a lot of research by scientists and biologists. Scientists have found that cancer is characterized by the uncontrollable growth of the body cells. In a healthy body, cell growth and differentiation is controlled and regulated. Normal cells display the property of contact inhibition, that is, the inhibition of uncontrolled cell growth due to their being in contact with other cells. In cancer cell, this property of contact inhibition is completely absent. As a result, cancerous cells divide to form abnormal masses of cell called as tumor. Tumors are either benign and malignant. Benign tumor cause little damage as they remain confined to their original location, do not spread to other part of the body. On the other hand, malignant tumor, also known as neoplastic cell, grow very rapidly and damage the surrounding normal tissue. They compete with other normal cells to provide a nutrient, thereby starving them. Cell passed off such malignant tumor travel to other parts of the body through the blood and start new tumors elsewhere. This frightening property of malignant tumors is also known as metastasis. Now, causes of cancer. The physical, chemical, and biological agents are responsible for transforming normal cells to cancerous neoplastic cells are known as carcinogens. Different types of ionizing radiations like X-rays, gamma rays, and non-ionizing radiations like UV rays cause DNA damage, leading to neoplastic transformation. Similarly, the chemical carcinogens nicotine present in tobacco causes lung cancer. Biological carcinogens can be found in virus called oncogenic virus as they can induce the formation of tumors. They are also called as tumor virus and have gene called viral oncogenes. Even normal cells have certain genes like cellular oncogenes and proto-oncogenes which when activated under certain conditions such as mutation led to oncogenic transformation of cells. This cell transformation in turn induces uncontrolled cell division due to this scary nature of proliferation of cancerous cells in an infected body. Of cancer is essential for its successful treatment. Cancer detection and diagnosis. Cancer is detected by using a variety of methods such as biopsy, histopathology of blood, as well as tissue and bone marrow test. In a biopsy, a piece of suspected tissue is cut into thin sections and is stained and examined under a microscope by a pathologist. This microscopic examination of tissue to study the manifestation of disease is called histopathology. Bone marrow tests are also conducted to detect increased cell count in the case of leukemia. Apart from these methods, detection of cancer in internal organ is made possible by techniques such as the radiography, that is, the use of X-rays, CT or computed tomography, and MRI, that is, the magnetic resonance imaging scan. The difference between CT scan and MRI is that the former uses X-rays to generate the 3D image of insides of an object, whereas the later uses a strong magnetic field and non-ionizing radiation to detect the pathological or physiological changes inside the tissue. Certain cancers are detected due to antibodies produced by the immune system against cancer-specific antigens. Cancer-specific antigens are proteins or other molecules that are unique to cancerous cells. These molecules are usually found in the outer plasma membrane and they are thought to be potential targets for the immunotherapy or other type of anti-cancer treatment. Molecular biology proved useful in identifying genes in an individual who are prone to inherit susceptibility to certain cancers. Identification of such genes is useful in preventing cancer since these individuals can be advised on avoiding specific carcinogens. For example, a person genetically susceptible to lung cancer can be advised to avoid a particular carcinogen like tobacco. Treatment. Cancer is usually treated by a combination of surgery, radiation therapy, and immunotherapy. In radiation therapy, tumor cells are irradiated lethally while taking into account not to damage normal surrounding cells. Sometimes cancerous cells are killed by chemotherapeutic drugs, which may be specific to particular tumor. Since the human immune system is unable to detect and destroy tumor cells, patients are administered substances like alpha interferons that activates the immune system and helps to destroy the tumor. These substances that activate the immune system are called biological response modifiers. In this manner, cancer can be diagnosed and treated with the help of several medicine techniques if detected at early stages. However, it is a killer disease and most often goes undetected by the human immune system.